Nisan, Jade, and Batislaon. In this video, I'll be showing you how I made my 3D model of an animal cell. This is with regards to my midterm performance task in Biology 1. So without further ado, let us start. For you to be guided all throughout the course of this video, this will be a sequence of how I am going to show you the parts of an animal cell. Starting with the cell membrane, followed by the cytoplasm, nucleus, nucleolus, mitochondria, vacuole, lysosome, vesicle, centrioles, your Golgi apparatus, ribosomes, and smooth and rough ER. The materials needed for this output includes the following: styrofoam balls, a large one and a small one, your paint brushes, a palette, your glue gun and glue stick, your cutting tools, a piece of cardboard, a folder, some beads, a pair of bottle cups, and your pencil. Now that we have all the materials, let's go! Cell membrane. It is the outer lining of the cell which encloses all the cell organelles. It also controls the movement of materials in or out of the cell. To make your cell membrane, cut your large styrofoam ball into half. Remember, in using your cutting tools, be extra careful. As you can see, I'm doing some trimmings. And there you have it. After this, color the styrofoam bowl with any color you like. In my case, I'll be using the color blue. And there you have it. Cytoplasm. It is a fluid substance that fills the cell. Thus, all organelles are suspended in it. For your cytoplasm, draw a circle on your folder by tracing your cell membrane. By using scissors, cut this, and there you have it. For my cytoplasm, I'll be using a lighter shade of blue. Okay, paint, and there you have it. Nucleus. It is a membrane-bound organelle that controls most of the activities in the cell. It stores the cell's genetic materials, or DNA. For your nucleus, using your cutter, cut the styrofoam ball into half. After this, using your cutter gun, cut the bottom part of the styrofoam ball. Be sure to keep it because we will use it later. After this, paint the styrofoam ball with any color you like. In my case, I'll be using purple. Or you can call it violet. There you have it. Nucleolus. It is an organelle found inside the nucleus. It aids in protein formation and RNA synthesis. For the nucleolus, we will be using the bottom part of the small styrofoam ball we used earlier. So cut it along the edges for it to be smaller. After this, color it with a darker shade of the color you used for the nucleus earlier. There you have it. Mitochondria, known as the powerhouse of the cell, where it produces most of the chemical energy for the cell. It is a double membrane organelle that has numerous folds and produces most of the ATP in the cell. For the mitochondria, we'll be using an excess styrofoam from the large styrofoam we used earlier. So using your cutter, cut it into shapes that resembles beans. Make sure that the top part is as flat as possible. So color it. I will be using the color orange for this part. Next, grab 2 pieces from your folder. Using your scissors, cut curvy shapes on it just like this. Color it. I'll be using the color yellow. And there you have it. After this, attach each part using your glue gun. There you have it. Next is the vacuole. It is a membrane-bound chamber that performs secretion, excretion, and storage inside the cell. For your vacuole, I'll be using these bottle cups. Cut this part. Then paint. I'll be using the color green. There you have it. Lysosomes. These are special vesicles that contain enzymes. They break down products and detoxify the cell. For this part, I'll be using the beads. Color it. I'll be using the color orange. Paint. And there you have it. Vesicles. They are small membrane sacs made up of fluid enclosed by a liquid bilayer. They transport materials within the cytoplasm. Just like the lysosome, I'll be using the beads for this part. I'll be coloring it with yellow-green. Paint. And there you have it. Centrioles. These are paired barrel-shaped organelles. They organize the microtubules, also known as a cell's skeletal system, and play a key role during cell division. For this part, grab a small piece from your cardboard. Cut a small strip from it. Cut it into half. Well, it depends on how thick you wanted your centrioles to be, so I'll be cutting it into half. Then, rip the top part of your cardboard so that the wavy part will be visible. This will allow us to mimic the appearance of the centrioles. So roll it, and using your glue gun, glue the end parts. Then paint it. I'll be using the color yellow. And there you have it. Next is Golgi apparatus, also known as Golgi body. It modifies the proteins made by the cell and pack and export proteins across the cell. For this part, I'll be using a piece from the folder. Using your pencil, draw shapes on it that resembles your Golgi body. Using your scissors, cut it. Don't think much about it. Just cut it and cut it and cut it. There you have it. Then paint it. I'll be using the color red. Voila! Ribosomes. These are the smallest yet most abundant cell organelles. They synthesize protein for the survival of a cell and are found on the endoplasmic reticulum. For this part, I'll be using the foam balls coming from your styrofoam, since these are considered to be the smallest. Color it. I'll be using the color yellow. And there you are. Last but not the least, endoplasmic reticulum. It is a large network of interconnecting membrane tunnels. It is divided into two parts, the rough endoplasmic reticulum, or RER, and smooth endoplasmic reticulum, or SER. The rough endoplasmic reticulum is covered with ribosomes, resulting to its rough appearance. This organelle plays a certain role in protein synthesis. The smooth endoplasmic reticulum, on the other hand, is not covered with ribosomes. It is involved in the synthesis and storage of lipids. For the endoplasmic reticulum, I'll be using a piece from the folder. Using my pencil, I'll be drawing shapes that resemble the endoplasmic reticulum. Using your scissors, cut it. So I cut it into half so that I'll have my rough endoplasmic reticulum and smooth endoplasmic reticulum. Afterwards, paint it. I'll be using the color orange, by the way. So as what is said earlier, the rough endoplasmic reticulum contains ribosomes, but in my case, I'll be putting the ribosomes later. So now that we're done creating the parts, let us now assemble it. So I'll be starting by sticking the cytoplasm on top of the cell membrane. After this, I'll glue the nucleus on top of the nucleus, followed by putting it onto the cytoplasm. then the endoplasmic reticulum, and the Golgi apparatus. Make sure that your endoplasmic reticulum and Golgi apparatus should always be near the nucleus. After this, I'll be putting the mitochondria, the vacuoles, your centrioles, followed by the vesicles. Oops. It was pretty hard putting the vesicles because they're so small. After this will be the lysosomes. And then your ribosomes. Voila! You now have your 3D model of an animal cell. According to Edward Abbey, you must learn to think not only logically, but also biologically. Again, I am Nisan, Jade, and Batislaon, and this is Biology 202.